series. Welcome to the RNA Collaborative Seminar Series. This is hosted by the RNA Institute at University at Albany, SUNY, and I'm going to be moderating. My name is Ken Halvorsen. I'm a senior research scientist here at the RNA Institute. The Collaborative Seminar Series incorporates, I think, up to 27 groups now, and we are one of them, one participating group, and sponsored by the RNA Society. I will note you don't need to, please don't take any pictures or audio. This will be recorded and it will be uploaded to the RNA Collaborative YouTube channel. On that channel, we have 86 seminars dating back to 2020, and within the next few days, this seminar will be added as well. So today we have two talks from people here at the RNA Institute. The first talk is going to be Arun Richard Chandrasekaran, who's a senior research scientist at the RNA Institute. And then our second talk will be Hannah Shorrock, who's a research scientist in the Berglund lab, also at the RNA Institute. And so we will kick things off with Arun, who's going to tell us about dynamic control of nucleic acid nanostructures using RNA and RNA degrading enzymes. You should be able to share, Arun.
Yep. You see it?
Yep. Take it away.
Right. Thanks. Hi, everyone. I'm Arun Chandrasekaran. I'm a senior research scientist here at the RNA Institute at Albany. So today I'll talk a bit about how we use RNA in nucleic acid nanostructures to make these structures more functional. So when we want to build things at the nanometer scale, there are two major approaches. The first approach is top-down, where you take a large chunk of a material and break it down to any shape or size you want, for example, like sculpting a statue. In the scientific realm, an example is lithography. The other approach is to take really small things, let's say atoms or molecules, and build it up to any shape or size you want. And in our case, the bricks that we use to construct are nucleic acids. And predominantly, we've been using DNA to construct such nanoscale structures. We are all familiar with the iconic double helical structure of DNA, and for simplicity, in this talk, I've represented them as two lines with base pairing in between. If we want to go more into detail of the structure, we can think of Daenerys Targaryen in Game of Thrones. And if you look closely, this picture has a mirror symmetry. If you slice it in the middle, the braid on her left shoulder is a right-handed double helix, and the braid on her right shoulder is a left-handed double helix. And for construction, we use the predominant pre-DNA structure that occurs in nature, although other transient forms are also used now and then. But why DNA? DNA is inherently a nanoscale material with a diameter of about 2 nanometers, a persistence length of about 50 nanometers or 150 base pairs. And it has a short structural repeat, so you get repeating units. And more importantly, the canonical base pairing of adenine and thymine and guanine and cytosine provides us with a programmable assembly where you can design specific DNA sequences that can bind to each other at will and how you dictate it. And these sequences can now be synthesized at really low cost and with a large library of chemical functionalities available. In order to build structures, we also need some kind of a mortar and not just bricks. In the case of DNA, we borrow from molecular biology a technique called sticky end cohesion. So you have two DNA double helices with a single stranded extension. For example, A here is complementary to A star. And now when you put these two molecules together, they cohere to form a pseudo-continuous duplex. You can now use such a technique to connect different duplexes. However, all you will be left with is a really, really long piece of DNA. So in order to create multi-dimensional objects, we need to literally branch out. And in order to do that, we create branched DNA junctions. We can design DNA with specific sequences. For example, sequence A in the light blue strand is complementary to A star in the dark blue. And same way, sequence D in the dark blue is complementary to D star in the orange strand. So when you now mix these 4 strands together and anneal them, you get the structure, which is a 4-way junction. The dark blue strand is now complementary to the light blue in the top left domain, and the strand crosses over to be complementary to the orange in the bottom right. And the same way, the dark red strand is complementary to the light blue in the bottom left, the strand crosses over and is complementary to the orange strand in the top right. So we call these points crossovers, and the 4-way junctions are also flexible. So you can imagine it in this conformation where it's a 180-degree turn. And we're all probably more familiar with a 4-way junction in this way, which also occurs in biology sometimes, for example, the Holiday junction. And over the years, different groups have created DNA junctions with different number of arms and used it for construction. And now you have both branch junctions and sticky end cohesion. And in order to create larger assemblies, you can now combine the two. You take a 4-arm junction, for example, and tail it with sticky ends. A is complementary to A star, B is complementary to B star, and now these molecules can connect to each other to form a quadrilateral. And in this case, you still have sticky ends on the outside, so this assembly can continue infinitely to form a 2D lattice. Here are some visual examples of DNA nanostructures people have created over the years. For example, a DNA origami smiley face, and more recently, a miniature Mona Lisa. So these examples show the extent of flexibility that DNA allows for constructing nanoscale objects and structures. In our lab, we focus on DNA nanotechnology, predominantly with DNA and some with RNA. And we mainly focus on functionalizing these nanostructures to make them better suitable for different applications. We focus on designing DNA crystals that can act as scaffolds for other gas molecules. We use DNA structure encoded information for data storage, and we're also building DNA nanostructures for drug delivery applications. And today I'll focus on the functionalization part and on how we incorporate RNA into nucleic acid nanostructures. And the reason we do that is having RNA single-stranded extensions can provide RNA toeholds, which can have a different functionality from DNA toeholds. DNA-RNA hybrids have different characteristics compared to an all-DNA or an all-RNA structure. RNA modifications can allow more functionality in these structures compared to just DNA modifications. And the incorporation of RNA also allows us to use RNA processing enzymes for structural reconfiguration and other purposes. And specifically, I'll talk about 2 projects that we have done recently. One is the use of 2' 5' linked RNA in nucleic acid nanostructures. And the second is where we developed a Toehold clipping strategy to control strand displacement. So I'll first kick off with using 2' 5' linked RNA in nucleic acid nanostructures. So typically, nucleic acids have 3' 5' linkages, and RNA is known to have 2' 5' linkages in nature. This can now be synthesized in-house for both DNA and RNA, although in nature, only RNA is known to occur. And the synthesis capability now allows us also to create oligos with mixed linkages, for example, both 3' and 5', to test how different number of linkages could have a role in both structural conformation and in functionality. To do this, we chose different model structures. Our first model structure was a simple duplex. Our second was a 4-way junction. Our third structure is a double crossover motif. It has 2 adjacent double helical domains connected by 2 crossover points, and hence the name double crossover. You can see from the model that the red strand runs through the helix in the top, and the dark blue runs through the helix at the bottom. The yellow and the orange strands sort of staple it together, and the blue strand runs through both the domains. And our final structure is the tensegrity triangle motif. It has 3 double helical edges, and in this case, each edge is 3 double helical turns or 31 base pairs. And the reason we chose these structures is that it allows us to test the incorporation of RNA 2' 5' linkages with increasing complexity of the structures. For example, the 4-way junction has a single crossover point, the DX has 2, and the tensegrity triangle has 3 branch points. Each vertex of the triangle is a 4-way junction. So we first assembled the duplex. We replaced 1 strand with RNA, and then we replaced the RNA with RNA containing different number of 2' 5' linkages, typically from 1 to 4 linkages. We first tested assembly using non-denaturing polyacrylamide gel electrophoresis, and we saw that incorporation of these linkages did not affect the structure that much. We also confirmed these studies using circular dichromosome spectra. We start off with a DNA shown in gray, and as we incorporate the RNA and RNA 2' 5' linkages, we can see the shift from the B form to the A form, which is expected because we are changing one strand to RNA. We next performed the same experiment for a 4-way junction. We confirmed assembly and tested it using CD spectra with the same results. And then we moved on to the double crossover motif. We again saw that assembly was not affected that much when we increased the number of linkages from 1 to 4. And we also confirmed it using CD spectra. And here we also performed UV melting experiments to see whether the incorporation of RNA or the incorporation of 2' 5' linkages affects the thermal stability of these structures. And we saw that incorporation does have a slight impact on the UV melting temperatures, but it's not that much when we increase the number of linkages. And when we moved on to the tensegrity triangle motif, we first started with a structure that is symmetric. Each edge of the triangle contains the same sequence, which means if we incorporate one strand of RNA with a modification here, we are incorporating that in all 3 edges. And what we saw that when we used this design, the assembly yield was affected, and we were not able to form the structures that well. So we changed to an asymmetric design where we formed the triangle with 7 unique strands. So the 3 double helical turns are now containing different sequences. So now we can change only one strand to RNA and then modify it to contain different number of linkages. We then assembled these structures, tested assembly with different number of linkages, and show that it has minimal impact on the assembly. And again, showed using CD that these structures move from B form to A form on the incorporation of RNA and 2' 5' linkages. We worked with Alan Chen at Albany Chemistry to also see how these linkages are tolerated in a duplex structure using molecular dynamic simulations. We simulated an all DNA structure and then a DNA RNA hybrid where there was no 2' linkages or 1 to 4 linkages. And what we observed was a change in the bending angle dynamics. Going from left to right, after an RNA strand is incorporated, you can see the distribution changing with higher number of linkages, which shows that there is some fluctuation in the structure once these modifications are introduced, and especially with higher number of modifications. So combined together, when these structures are put together on the centroid axis, you can see that there is some fluctuation in the bending angle. However, the overall structure seems to tolerate up to 4 linkages that we tested here. And after these characterizations, we then wanted to see how these structures can be useful in nucleic acid nanotechnology. So one aspect of 2' 5' linked RNA that is already known is that these modifications can resist nuclease degradation by RNases. And that's also why they are starting to be used in antisense therapeutics. So in our experiment, we took a native DNA RNA hybrid and a DNA RNA hybrid that contained 4 2' 5' linkages. We then added RNase H, which can cleave an RNA in a DNA RNA hybrid. And what we saw with increasing amounts of the enzyme was that the 2' 5' linked structure was slightly more stable, that is more biostable compared to the native structure. We then performed a time series to see the degradation over time. And again, we saw that the structure containing 2' 5' linkages was more biostable than the native structure. And this shows an important feature that can be achieved using such modifications in creating nanostructures with enhanced biostability. To summarize, this was the first report that incorporated RNA strands containing 2' 5' linkages into nucleic acid nanostructures. And we demonstrated using 4 different structures that these structures tolerate these modifications to different extents. And based on the nuclease-resistant experiments with RNases, we see potential that these structures be useful in drug delivery, where drug delivery carriers are expected to be more stable until it reaches the target site. I'll now discuss about our project on Toehold clipping, where we controlled strand displacement processes in nucleic acids. Over the years, there have been several strategies that are used to control DNA nanostructures, or nucleic acid structures in general. For example, you can use pH to control the reconfiguration of a structure. For example, you can use triplexes or I-motifs that are pH sensitive. In some cases, you can use temperature to open or close the structures to release certain moieties. And you can also incorporate photoclebal linkers to create light-triggered response. And another example is antigen aptamer or antibody aptamer interactions. And finally, nucleic acids that can remove other sets of nucleic acids from these structures, a process called strand displacement. And we focus on strand displacement and have the question whether we can add an additional layer of control using light or enzymes. Before I discuss these two strategies, I'll give a brief overview of what Toehold mediated strand displacement is. So if we start with a duplex with a single-stranded extension, the duplex portion has the sequence B and B star that are complementary, and the single-stranded region has a sequence A, which we call a toehold. If you now add another strand of sequence A star and B star, which we call an invading strand, since A star is complementary to A, this strand now binds to this toehold region, sort of stepping a toe on the ledge. And then since the rest of it is complementary to the lower strand, it continues to bind and eventually kicks out the short strand. And if you want to remember this process better, you can think of the song Break Free by Ariana Grande, where the lyrics go, this is the part when I say I don't want you, I'm stronger than I have been before, and this is the part when the longer strand breaks free because it can't resist no more. So in our case, what we did was to incorporate a photoclebal linker in between the duplex domain and the toehold. And now strand displacement should continue as usual if there is no stimuli. However, once you shine UV, the photoclebal linker is cleaved, the toehold is now cut off. And now if an invading strand is added, this duplex remains the same and strand displacement does not happen because once there is no toehold, strand displacement process becomes much, much slower. And the way it works is we synthesized a DNA strand with a photoclebal linker that is commercially available. We have the strand continuing to the duplex on this side and the toehold on the right side. And in the presence of UV, the strand gets cleaved. We first tested to make sure that UV triggered cut is actually happening. So we took the single strand, which is a 29 mer, and in the absence of UV, you can see that the 29 mer is intact. And in the presence of UV, the strand is cleaved at the photoclebal linker, and now you get a 21 nucleotide product, which confirms that we can use UV to cleave the strand. We also monitored the time that it takes for the strand to be cleaved, and at about half an hour to 1 hour, we get almost 100% cleavage of the strand with the toehold being removed. So now that we know it works well in single strands, we then tested whether toehold clipping also works in duplexes. So we put together the duplex, and for a DNA toehold, you can see that UV does not cause any impact. However, for a duplex with a photoclebal toehold, you can see that this is a larger duplex, and on the UV exposure, the toehold is cut off, and you're now left with a shorter duplex. We then moved on to see whether we can use toehold clipping to control strand displacement in these duplexes. So as our control system, we have an all DNA system. You start with a duplex with a toehold, and in the presence of an invading strand, the short strand is kicked off, and you end up with a longer duplex, which is observed as a slower moving band on the gel. And you now have a photoclebal linker toehold, and in the absence of UV, you can see you start with a duplex with a single stranded extension, and strand displacement happens, yielding a longer duplex. However, on the exposure of UV, toehold displacement is now affected, and we quantified these results, and we can curtail DNA strand displacement by up to 60% by using photoclebal linkers. And next, we tried to see if we can do a similar process, but using RNA and RNA degrading enzymes. So here we incorporated an RNA toehold, and I would like to stress that the lower strand here is a DNA RNA chimera. The B sequence shown in red is DNA, and the sequence A in blue is RNA. So now if you add an invading strand, again, strand displacement happens and the shorter strand is kicked off. However, when you add RNase A, which is specific to single-stranded RNA, only the toehold portion in this complex is digested, And since the remaining portion is all DNA, the duplex is left intact. And now if you add an invading strand, there is no toehold to bind to, and strand displacement does not occur. Again, we tested this in a single strand first. You start with the 29-mer, where it's part DNA and part RNA. On the addition of RNase A, the toehold is digested and you're left with the 21 nucleotide DNA. And we confirm that this can also occur in duplexes. You start with a duplex with a single-stranded extension. On the addition of RNase A, the toehold is cleaved and you're left the shorter duplex. We then moved on to see if we can use RNase-controlled toehold clipping to prevent strand displacement. So in a DNA control, you can see that you start with a duplex and strand displacement occurs. Same with an RNase toehold without any RNase added to it. But once you add RNase A, you're starting off with a duplex, and you can see that toehold clipping prevents strand displacement to a large extent, and we are able to control or prevent strand displacement by about 85% when we have an RNA toehold and clip it using RNase A. Now that we have shown the toehold clipping strategy in duplexes, we then wanted to see if we can use this strategy in more complex DNA nanostructures. So we chose to work with the 3-point star motif, which has 3 arms emanating from the center. Each arm of the 3-point star motif contains 2 double-stranded domains connected by 1 crossover point in the middle. And this motif is 3-fold symmetric, so each arm is containing the same sequence. You have a short strand shown in red on all 3 edges, and then you have a medium strand shown in blue, and then a long orange strand that runs through in the middle. And this shows a molecular model of the structure. And in each arm, there is also a sticky end that allows these motifs to connect to each other. And past the sticky end, there is a single-stranded toe that we incorporated. And we chose this motif because in assembly, we can control hierarchical assembly where 4 such motifs can connect via sticky end to form a closed object, which is a DNA tetrahedron. And in each arm, there is a toehold that we can use to displace the short strand and make the tetrahedron disassemble into its component parts. If we take a zoom in into this process, the tetrahedron is formed by 4 such units that are connected by 2 sticky ends on each edge. And past the sticky end, we designed a toehold that is coming out of the center. And once you add an invading strand, it binds to the toehold. It continues to bind to the rest of the short strand and then removes it from the structure. And now, since there is no sticky end, the tetrahedron falls apart into its starting units. So the idea is, can we use toehold clipping to prevent this disassembly and keep the tetrahedron intact, even in the presence of an invading strand? So first, we assemble the tetrahedra, make sure we can form the tetrahedra. We had our controls, which are individual strands, and then the motif itself. And then we made a tetrahedron without any toehold, a tetrahedron with a DNA toehold, a tetrahedron with a toehold containing a photocleavable linker, and finally, a tetrahedron containing an RNA toehold. And we see that we can form all of these structures pretty well. An earlier group also showed cryo-EM structures of these DNA tetrahedra, and you can see that it's a pretty defined structure that is hollow. Next, we wanted to see if we can actually do toehold-based strand displacement and disassemble the tetrahedron. And if you look at these lanes, first, when we have an all DNA structure, you see the tetrahedron band, and then in the addition of an invading strand, the tetrahedron is disassembled into its component motifs. And we can see the same trend in a DNA with a PCL toehold and an RNA toehold, where displacement causes the tetrahedron to disassemble, and this band now goes down in intensity. However, in a DNA tetrahedron without a toehold, you can see that even the addition of an invading strand does not have any effect. So this shows that disassembly of the tetrahedra is definitely because of the toehold, and further we can control it. And next, we moved on to see whether we can use light or RNAs to control the strand displacement process, and in turn control disassembly of the structure. So we start off with the PCL toehold, where we have a photocleavable linker. In the absence of any UV light, we start off with the tetrahedron, and in the addition of an invading strand, we see that the tetrahedra is disassembled with about 90% disassembly. However, on the addition of a UV stimuli, the toehold is now clipped, and on the addition of an invading strand, the tetrahedra remains intact. And with the RNA toehold, without an RNase present, the tetrahedron disassembles on the addition of an invading strand. However, when you add RNase A, the toehold is clipped, and now since there is no toehold, the tetrahedron remains intact even in the presence of an invading strand. So this shows that we can not just control strand displacement processes in single strands and duplexes, but also in complex nanostructures. To summarize, we've shown that we can introduce an additional layer of control over strand displacement reactions using both light and RNAs by incorporating toeholds that contain either a photocleavable linker or by incorporating an RNA toehold. And this strategy is not just applicable for duplexes, but it can be incorporated into larger and more complex DNA nanostructures, even those that require hierarchical assembly. So we see that these strategies have potential in actuating devices for biosensing. For example, the invading strand could be a biological marker that then causes a signal readout, or in drug delivery, where a drug carrier could be intact until it finds a particular biomarker, which then causes the structure to disassemble and then release the drug. This paper was recently published, and we also got the cover in NAR. And with that, I conclude my science session, and I would like to thank my team. We are a newish lab. We started in June 2022, so we've been active for just over a year. So we are a growing group. I would also like to thank my funding agencies and the RNA Institute and Albany for startup funds. If you have any questions or if you want to chat, feel free to reach out anytime. If you're interested to work with us, again, reach out to me with any of these contacts. Thanks.
All right, thank you, Arun. Oh, I forgot to mention earlier that if people have questions, you can use the chat or the Q&A section. And I'll read aloud the first question, which is up there, which is from Subodh Mishra. What is the minimum number of base pairs to form a multi-core stable structure? And this was asked early in the talk. I think it was related to some of those heavily crossed over structures. You're on mute, Arun.
If the question is about a structure like this, so it comes down to each domain with the crossover. So when it comes to four-arm junctions, for example, we have a minimum length of 16 per arm for the junction to remain stable. For longer structure, we typically go in n number of turns. And again, for a structure to be flexible, it has to be at least 2 or 3 turns. The stability then has several different parameters. For structures to be able to assemble, each arm across the crossover should be about 10 to 12 base pairs.
Okay, we also have a live question here. I'll try to see if I can unmute Marcin Magnus for his question. Let's see if I know how to do this. Hold on.
I have given him the permission, so he can unmute and ask the question.
Oh, Marcin, unmute yourself and ask your question. All right. In the meanwhile, I'll ask.
There's one more on the chat.
Yeah. So you see that one? It says, does UV light also create thymine dimers that could impact the structure or further functionalization?
In these structures, we don't think that happens. In a different study, we have tested that by intentionally incorporating a few adjacent thymines. So unless they're really close thymines with some type of a cross-linker, these dimers don't usually occur, at least in the timeframes that we shine UV on.
All right, Marcin, if you're still interested in asking a question, you can unmute yourself and ask the question. Okay. Well, then, if not, I guess we will move on. Marcin, if you still have a question, you can still ask it in the Q&A, and Arun can respond to it offline. Sorry, we didn't get to your question. We are going to move on to Hannah Shorrock, who's giving the second talk on alternative splicing dysregulation in CAG expansion, Spinocerebellar Ataxias. So go ahead, Hannah, we see your screen.
Perfect, thank you. So good afternoon, everyone. And as Ken said, I'm going to talk to you today about alternative splicing dysregulation in CAG expansion and Spinocerebellar Ataxias. So Spinocerebellar Ataxias are a group of more than 40 autosomal dominant neurodegenerative diseases. And these are characterized by progressive ataxia. So that's a loss of balance and coordination that's accompanied by slurred speech. And this is primarily driven by atrophy of the cerebellum. So you can see that the cerebellum highlighted in red in the schematics of each of these subtypes of the scars are areas where we have a lot of atrophy in these diseases. And this is largely associated with damage to the cerebellar Purkinje neurons. But as you can see, other regions such as the brainstem are also frequently affected in the spinocerebellar ataxias. Now, spinocerebellar ataxias are genetically heterogeneous, and so the kind of more than 40 different types are caused by a different mutation within a different gene for each subtype. And the most common type of mutation that we see in this causes some of the most common of the spinocerebellar ataxia subtypes are those caused by CAG repeat expansion mutations. So, for example, a CAG repeat expansion within a coding exon of ataxin 1 is causative for spinocerebellar ataxia type 1. And this is the case for many subtypes of spinocerebellar ataxias, where a CAG repeat expansion within a coding exon causes that disease. And for SCA12 here, we have a CAG repeat expansion that causes the disease, and this is located within the 5' UTR. Now, together, the estimated prevalence for the spinocerebellar ataxias is about 2 to 6 per 100,000. So these are generally quite rare diseases. And when we think about this repeat expansion a little bit more, you can see that each different disease-causing gene has a different threshold that causes the disease. So, for example, if you had, say, 15 repeats within the ataxin 2 gene, you wouldn't develop spinocerebellar ataxia type 2. But if you had 40 or 50, say, this would be above the threshold that would become pathogenic and would develop spinocerebellar ataxia type 2. As I said earlier, the primary symptoms are progressive ataxia across all of these subtypes of spinocerebellar ataxia, but you can see as well that individual subtypes here do have distinct features as well. When we think about the disease mechanisms for these CAG expansion scars, we have this CAG repeat expansion that's located within the disease gene. And we know that this is transcribed into a CAG containing RNA, which then gets translated into polyglutamine proteins in the majority of the CAG scars. There's been a lot of work studying these polyglutamine containing proteins, and we know that these aggregate, and this can lead to impaired proteostasis in the cells. There are a variety of other downstream pathways that are affected in these diseases, including things like altered DNA repair, perturbed neuronal circuitry, and impaired bioenergetics. And we can look at this in a different way as well. So here you can see the polyglutamine protein aggregation. But as I said, there's a variety of other cellular pathways affected, including ion channel dysfunction, disruption of membrane conductance, transcriptional dysregulation, DNA damage, and a variety of other nuclear processes as well. Now, despite knowing that a large array of cellular pathways are particularly affected in the CAG scars, the precise underlying molecular mechanism is unknown for any spinocerebellar ataxia. So I'm particularly interested in understanding overlapping mechanisms of disease pathogenesis and working out how we can leverage this for cross-disease biomarker and cross-disease therapy development. And so I was really struck by there being kind of a lack of consensus of particularly pre-symptomatic disease-driving transcriptomic changes in the CAG expansion scars. And so I was really interested to perform a comprehensive analysis of RNA sequencing datasets trying to see what consensus I could see within RNA sequencing data sets from across CAG expansion scar mouse models from scars 1, 3, and 7. And so while this work started off looking at differential gene expression, it actually rapidly turned into a study of alternative splicing in the Spinocerebellar Ataxias. So these CAG expansion scars belong to a much larger family of diseases called microsatellite expansion diseases. And when we think about dysregulation of alternative splicing across microsatellite diseases, it's something that's been reasonably well studied in other diseases. So recently, there's been several reports of dysregulation of alternative splicing in the CAG repeat expansion disease, Huntington's disease. But most notably and very well studied is that in myotonic dystrophy, we know that misspliced events have been linked to key phenotypes. So myotonic dystrophy type 1 is caused by a CTG repeat expansion. And when the CTG repeat expansion is transcribed into RNA, we have these CUG expansion RNAs that sequester the MBNL family of RNA binding proteins. And because the MBNL is bound to this RNA, we then get dysregulation of MBNL-regulated alternative splicing events. So you can see one example here. So this is a chloride ion channel. And normally in adults, we have exclusion of this exon 7A, and this leads to the production of full-length and functional chloride ion channel proteins. When MBNL is not able to perform its alternative splicing regulation in DM, what we see is inclusion of this exon 7A, which has this premature stop codon. And so this leads to nonsense-mediated decay, which leads to a reduction in chloride ion channel conductance and the hallmark myotonia that's characteristic of myotonic dystrophy. And so this is a really nice, clear example that dysregulation of key alternative splicing events can directly cause symptoms in myotonic dystrophy. And so this is a very different landscape from what we know for the CAG expansion scars. So as I said earlier, precise underlying molecular mechanism is unknown for any scar. And we know that RNA-mediated disease mechanisms are understudied in the CAG expansion spinocerebellar ataxias. So to be able to form this comprehensive analysis of RNA sequencing data sets, we first had to identify the RNA sequencing data sets that we were going to use. So we set several criteria and formed a comprehensive search for data sets, and those that passed our read depth threshold for being able to analyze alternative splicing are the ones shown in black here, and all those data sets are included in our study. So here I'm showing you our alternative splicing analysis using RMAT across 13 different data sets from the cerebellum. And these come from 2 different SCAR1 mouse models, so one SCAR1 mouse model there and another here, 2 different SCAR3 mouse models, and then 1 SCAR7 mouse model. And what you can see across all of these data sets is that we do see dysregulation of alternative splicing, with the most impacted event type being skipped or cassette exon events, and they're shown in the dark blue here, with the number of events detected in each data set highlighted. When we look at the magnitude of those changes in alternative splicing, you can see that we're not just looking at small changes. So some of the events that we're looking at here have quite sizable magnitudes in the change of the splicing that we're seeing between the wild type mice and these CAG scar mice. We were also interested to understand the extent to which these dysregulated alternative splicing events were shared between these different data sets. And so while you can see that across these data sets in dark blue here, the majority of events are generally present within only that data set, all the data sets contain events that are shared between at least 2 or more other data sets. And so you can see when we look at that overall, we have over 500 events that are shared between at least 2 different data sets throughout the cerebellum. When we look at the events that are shared between 4 or more data sets, and we use them to look at each data set individually, you can see that these events separate out our wild type mice, which are shown in circles here, and our SCA mice, which are shown in triangles. And so you can see very nicely from this gray data set that this occurs kind of at the level of the individual data set, but more globally, these events are capable of separating out the scar and the wild type mice across these data sets. And you can see more triangles over this side of the PCA plot and more circles over here. We're also interested to understand whether the skipped exon events were occurring in disease-relevant pathways. And so here we're looking at a gene ontology enrichment analysis of those skipped exon events that were shared between 2 or more of these cerebellar data sets. And what you can see here is that these skipped exon events are occurring in genes that are relevant for CAG expansion scars. So for example, things like regulation of synapse organization, actin filament-based processes, regulation of membrane potential, and splicing factor nova-regulated synaptic proteins. And we can look at this in a different way here. So here, each color represents an individual data set, with each point representing a single gene ontology enrichment term. And again, you can see that we're seeing the same kind of terms coming out. So we're seeing enrichment of terms associated with the cytoskeleton, with synapse structure and function, and with ion channels, as well as more generally with a variety of nuclear processes. Because we know that these diseases are caused by repeat expansions when they're over a particular threshold, we're interested to understand whether these changes that we're seeing in splicing are repeat length dependent. And so for 3 of these RNA sequencing data sets, mouse models were generated using exactly the same strategy, but containing repeats within the normal repeat length. And so here we've compared the pathogenic repeat expansion mouse models with their short repeat counterparts. And so here we have this analysis for 1 SCAR1 data set and 2 different SCAR3 data sets. And when we do this analysis, what we saw was that we're actually seeing a similar number of skipped exon events being dysregulated, as we saw when we compared these data sets to wild-type mice. And what's more is that the magnitude of changes that we were seeing were comparable to the magnitude of the changes that we saw when we were comparing these to wild-type mice. So this really suggested to us that these changes are repeat length dependent. We were also interested to understand the interplay between differential gene expression and alternative splicing dysregulation. And so we looked at the number of differentially expressed genes and skipped exons within these repeat length comparison data sets. And what you can see for this pre-symptomatic SCA1 data set and this early symptomatic SCA3 data set is that we have far more skipped exon events than we do differentially expressed genes. But by the time we're at this late symptomatic SCA3 time point, you can see that we have a lot more differentially expressed genes. And so we were particularly interested to understand if skipped exon events perhaps preceded changes in differential gene expression, and if what we saw with this SCAR1 and SCAR3 data sets was something that was consistent across the pre-symptomatic data sets in this study. And so what we found was that across the board, in these pre-symptomatic data sets, we did see more skipped exon events being dysregulated than differentially expressed genes. And this was particularly prominent for this SCA3 data set here, and also for this SCA1 data set, which is by far the highest read depth in our study and gives us the best ability to be able to robustly detect skipped exon events. When we looked at the gene ontology enrichment analysis for the overlapping events across these 4 pre-symptomatic SCAR1 data sets, again, we saw terms that were really relevant for CAG expansion scars. So things such as splicing factor nova-regulated synaptic proteins, response to calcium ion, and synaptic signaling. So now that we knew that we were seeing alternative splicing dysregulation pre-symptomatically and in disease-relevant pathways, we were interested to track individual events across disease and to begin to understand what the individual event regulation looked like. So here I'm showing you an example of an ion channel TRPC3. And what you can see here with the wild-type mice in dark blue and the short repeat mice in pale orange is that typically we see this exon 9 being included between 20 and 40% of the time in wild-type mice within the cerebellum. But when we look in cortical and brainstem regions here, you can see that this exon is included almost 100% of the time across the board. And so what we're looking at here is a cerebellar-specific alternative splicing event. And what you can see in the CAG scar mice in the cerebellum is that we're actually seeing a shift of this exon 9 being included more. And this is moving the splicing profile of these TRPC3 away from what we would normally see in the cerebellum to what we would normally see in other brain regions. And this is something that we were able to validate using RT-PCR within an SCA1 mouse model. And so this particular cerebellar-specific alternative splicing has been well studied. And so we know that when exon 9 inclusion is increased, this actually leads to a reduction in membrane conductance. Here I'm showing you another example of another ion channel, KCNMA1. And again, within the cerebellum, we can see that in the wild-type mice and short repeat mouse, this exon is included maybe 10 to 30% of the time. But in cortical and brainstem regions, it's included more than 80% of the time across wild-type, short repeat, and scar mice. When we look in the cerebellum with the scar mice, again, we're seeing a shift away from what it should look like in the cerebellum to what it is in other brain regions. And again, we were able to validate this using RT-PCR. Again, this particular splicing event has also been well studied. And so we know that increased exon 23B inclusion actually leads to reduced channel activation. And so just to provide you with a little bit more context for those two splicing events, what you can see here, both TRPC3 and KCNMA1, is that the region that's affected by this alternative splicing is highlighted here in red or orange. And so we know for TRPC3, for example, in the wild-type mice without exon 9, what we have here is an incomplete CIRB motif. And with the scar mice, we have this complete CIRB motif, and this changes the permeability of those ion channels. And so this is how this leads to a reduction in membrane conductance. Similarly, this exon 23B here impacts this RCK2 domain calcium ball. And so again, this leads to alterations in the activation of the channel and changes in potassium ion channel efflux there. And so these are two examples that can give you an idea as to how alternative splicing of specific events in the CAG expansion scars could directly impact cellular pathways that we know to be disrupted and affected in these diseases. So what I've shown you so far is that alternative splicing dysregulation occurs pre-symptomatically in disease-relevant pathways and in a repeat length-dependent manner across SCAR1, 3, and 7 mouse models that contain these CAG repeat expansions. I've also shown you that we've been able to identify alternative splicing of ion channels with known functional consequences, but many of these changes are actually also occurring in genes with pathogenic links to spinocerebellar ataxias. So for example, we know that mutations in TRPC3 cause SCAR41, and mutations in KCNMA1 cause a progressive form of ataxia. And we identified dysregulation of alternative splicing events of many genes that are known to cause different subtypes of spinocerebellar ataxias. So now that we've identified that alternative splicing is a transcriptomic hallmark of these diseases, we're really interested to understand whether it can be used to assess therapeutic efficacy in CAG scars. And when we think about therapeutic strategies in these diseases, clinical care does currently focus on managing symptoms, and preclinical therapy development largely focuses on disease-specific approaches. So for example, one area that has a lot of preclinical development is using antisense oligonucleotides that would target the disease-specific transcript to be able to reduce the overall burden of these CAG expansion transcripts. There are also therapeutic strategies that target general neuroprotective pathways, so targeting some of these downstream aspects of the disease here. And something that I'd just like to highlight at this point is that there's no small molecules published to reduce levels of CAG expansion transcripts. So several of the studies that of the data sets that we included in this study did include RNA sequencing for therapeutic intervention in these mouse models. And so what I'm showing you here is analysis that we did where we looked at the number of significant skipped exon events between the wild type and the untreated or sham-treated scar mice. And then we looked to see how those events were affected in the treated mice. And so what you can see here is that for this SCA1 ASO study from the cerebellum of these mice, you can see that the majority of the events here are rescued to a certain extent. We do have a large proportion of events here that aren't rescued or that perhaps even show a worsening of their degree of change. And you can see across the board here that most of these data sets do have more events that are rescued and then the number of events that aren't rescued or that show an opposite effect. We can also look at rescue of alternative splicing using a couple of different approaches. So for example, here, we can look at a more generalized view of this alternative splicing analysis by doing the PCA plot of the skipped exon events that are significantly different between the wild type and the untreated mice. And so here you can see that these SCAR1 treated mice treated with this ASO sit between the untreated and the wild type mice, indicating that there's a degree of rescue here. And we can also look more specifically at individual events. So here you can see that with this ASO treatment in these SCAR1 mice, these two events that we were able to validate in our SCA1 mouse model were rescued by this particular treatment. Now, something that I'm particularly interested in alongside my work looking at alternative splicing is identifying CAG expansion regulating small molecules that could potentially be used across multiple different types of CAG scars. And so we identified a compound that I'm referring to as HIT2 from CAG expansion selective small molecule screen. And here you can see that when we treated an SCA1 mouse model with this, we saw a 20% reduction of HIT2, a 20% reduction of ataxin-1 expression within the cerebellum of these mice. Now, we were really interested to assess whether this small molecule was capable of rescuing splicing in these mice. And so we performed RNA sequencing on the cerebellum of these mice. And before I get into the rescue data, I just want to show you that with our in-house RNA sequencing, using an RNA depletion pipeline, we were able to validate the previously published data sets that we analyzed, and we found the majority of the events from our in-house library prep. We saw dysregulation of skipped exon events here. And again, these were occurring in disease-relevant pathways. So we saw terms such as nuclear localization, regulation of synapse organization, and pre-symptomatic depolarization and calcium channel opening, for example, as disease-relevant terms that came out of this analysis. When we look at these 495 events, we were interested to understand whether they're rescued with the HIT2 treatment. And so what you can see from this PCA plot here is that we're seeing our HIT2 treated mice are sitting between our wild type and our DMSO treated mice, indicating that we do have reasonable rescue of splicing with this small molecule treatment. And when we look at this by the number of events that are rescued a certain proportion versus not rescued, you can see that the vast majority of events here are rescued. And when you think back to the graph that I showed you previously, where we looked at this metric across a variety of different data sets, the ratio that we see here for the rescue of splicing is actually a much better rescue ratio than what was seen for those other data sets that we analyzed. We can look at these rescued events in another way as well. So here we're looking at events that are significantly rescued between the HIT2 and the DMSO treated SCA1 mice. And what you can see is that the pattern of splicing in the wild type mice and these HIT2 treated mice are much more similar than they are to the DMSO treated mice in the middle here. And here I'm just showing you three individual events that were rescued with RT-PCR validation of CACNA2D2, which is another ion channel of interest. Okay, so what I've shown you so far then is that dysregulation of alternative splicing represents a novel transcriptomic hallmark in these diseases. It occurs pre-symptomatically, which is really important because we previously struggled to identify disease-driving transcriptomic changes in these diseases. And we've also seen that it affects genes and pathways implicated in the scars. I've also shown you that alternative splicing dysregulation represents a target engagement biomarker. So we can use both global and event-specific splicing dysregulation metrics to assess therapeutic approaches and how well they can rescue those events. I've also shown you that I've identified a novel CAG repeat selective small molecule that reduces expression of CAG expansion transcripts in an SCA1 mouse model. And this also actually reduces levels of CAG expansion transcripts in SCAR1, 3, and 7 patient-derived fibroblasts. And I've shown you that this compound rescues alternative splicing dysregulation in the cerebellum of SCA1 mice. And really where we want to take this is to understand whether this is relevant in patient models of these diseases. So everything that I've shown you so far has been looking at mouse models of these diseases. And so a wonderful graduate student in the lab, Asma Alieva, has been performing RNA sequencing of 3SCAR1, 2SCAR3, and 1SCAR7 patient-derived fibroblast lines. And what you can see here across the board is that we do see a large amount of dysregulated alternative splicing events, with that predominantly affecting skip exon events largely across the board. And again, when we looked at gene ontology enrichment of these skip exon events, disease-relevant pathways were highlighted, things like microtubule-based transport, chromatin organization, and cellular response to DNA damage stimulus. And so this is something that we're actively following up, and we're moving towards being able to use events that were detected in these fibroblasts to assess whether our lead candidate small molecules in the lab are rescuing those splicing events. And so with that, I'd like to thank my mentor, Dr. Berglund, as well as many of the trainees and staff who I've had the pleasure of working with on these projects in the lab. Our collaborator, Damien Shin at Albany Medical College, who we do our SCA1 mouse work with, and then of course our funding sources. And I'd like to thank you for taking the time to listen to me today, and I'll take any questions.
All right, thanks, Hannah. If anybody has questions, you can ask them in the Q&A or raise your hand. There's nothing yet. I'll ask a quick question, and maybe I just missed it. Do you have an idea of what the small molecule is actually doing structurally, or not yet?
So that's a really good question. That's something that we're looking into exactly what it's doing. So it's identified from a screen where we looked at expression levels. So it was identified because it selectively reduces expression of the CAG expansion transcripts relative to a zero repeat control. So we weren't particularly looking for things that bind the CAG repeat, for example. So we're currently looking into what the mechanism of action is. It could be kind of targeting a pathway that regulates expression of CAGs, or it could be directly targeting the CAG repeats themselves. Yeah.
All right. Well, here's a question. Great talk, Hannah. Are you using both male and female mice in testing your HIT2 treatment? From Carly McCumber.
Yes, so the study set out to use male and female mice. Unfortunately, there was a genotyping error, which resulted in not sufficient N numbers of female mice to include in the study in the end. So the data that I've shown you today is all for male mice, but that is something that we're working towards in future. And we're thinking of moving our timeframe for our initial studies as well. So that study was performed from 18 to 20 weeks of age. But due to various metrics that we can't assess at that age, we're thinking of moving our initial studies to an earlier time point in the mice.
One more question from Junji Guo. Do you think that the non-CAG SCAs may also show similar changes?
I think that's really interesting, and that's something that we're trying to look into with some of the fibroblast studies. So what I showed you with the mouse data is that we see missplicing of many of the genes that cause non-CAG scars. I am not aware of any publicly available RNA sequencing data to be able to directly assess your question, but I think we are interested to understand, is this a CAG-driven mechanism of alternative splicing dysregulation, or is there kind of more of an interplay between these genes that cause these very similar types of diseases other than just the CAG scars? So I think that's definitely an interesting question, something that we're looking into. Thank you.
Last chance for questions for Hannah. I guess if there's no further questions, we can wrap up and close the seminar. And thank both our speakers, Arun and Hannah, for great talks. Sangeetha, is there anything else I need to mention